This is thin skin. Thin skin means that we have skin. Skin consists of epidermis, dermis, and subcutis, 3 layers. Skin where the stratum corneum, the uppermost layer of the epidermis, the layer that does not contain any nuclei, is thinner than the rest of the epidermis here. As I said, the skin consists of 3 parts. We have to understand that if we are discussing skin. And the 3 parts are the epidermis, that's the epithelial part here, the epithelium, the dermis, which is the connective tissue, some looks pinkish here, and then the subcutaneous tissue where we mostly see fat. We will start to discuss the skin from top to bottom. Always, even if I'm discussing pathology slides in clinical practice, then I'm discussing the changes from top to bottom. It's important then one has a certain type of approach. But so we start with epidermis. The epidermis in skin is keratinized stratified squamous epithelium. That we have squamous epithelial cells that make up the top layer, and stratification, that means we have different layers. We have a couple of layers of epithelium, so we call it stratified, and we call it stratified squamous because we always name the type of epithelium according to the most upper cell. And then in this case it's keratinized because we have this thick keratin layer here. Actually, what happens in the skin is that the cells proliferate here from the basal layer. They will travel upwards and they will mature as they travel upwards. They will become more cuboidal, they will become squamous, and then they will lose their nucleus and become this keratin layer. So this is how this keratin comes about. Thin skin, if we speak about thin skin we have actually everywhere at the body except for areas that we have thick skin, and thick skin we have at the palms and at the soles. Basically there that we need a thick protective layer of skin. The basal layer here of the epidermis is known as stratum basale. Stratum basale consists of columnar epithelial cells. And interspersed in those columnar epithelial cells are also other types of cells, and those are mainly melanocytes. Usually we have 1 melanocyte per 10 to 20 keratinocytes. Keratinocytes are all the other cells here. This, for example, is a melanocyte. And then we have other cells like, for example, Merkel cells or dendritic cells, aka Langerhans cells. If we speak about melanocytes are very important in clinical practice because melanocytes can give rise to a couple of lesions, nevi, for example. So it's very important that you recognize what a melanocyte looks like, and the melanocyte looks grayish, more white, and not as pink as the keratinocytes. And then we have a little vacuole around the outside of the cell, which is actually an artifact which occurs due to tissue processing. I think this also might be a melanocyte. We have a more grayish cytoplasm and then a white kind of vacuole around the cell. And this occurs due to tissue processing. This is, of course, not how the tissue looks like when it comes out of the skin, but we process tissue in different ways, so it looks like this. And there are certain changes that can occur to the cell that will lead to the fact that it looks like this, all pink and purple when it comes out. The next layer that we have is actually the stratum spinosum. Stratum spinosum are those cells here in the middle, basically. And stratum spinosum, if we could go in more, which we can't, I think, then we could see that stratum spinosum cells, they're a little cuboidal, and they're connected here where you can see the white spaces through intercellular connections, and those intercellular connections are known as desmosomes. Those are bridges. They look a bit like ladders if you zoom in even more, and those ladders made these layer receive the name prickle cell layer because they appear prickly. So stratum spinosum, or the middle layer of the epidermis, is also known as the prickle cell layer, and the cells are connected via desmosomes. Why is this important? This is important because we can have certain autoimmune diseases where the body produces autoantibodies against a protein which is in those desmosomes, and then those connections are destructed, and then we have vesicles occurring here inside of the epidermis, intraepidermal, and this disease, if we have autoantibodies against a protein of desmosome, is called pemphigus vulgaris. Maybe you've heard of this. We have also another disease which is bullous pemphigoid, and bullous pemphigoid also affects an anchoring protein, so to say, and that is the anchoring protein that anchors the basal cell layer here to the basement membrane, because of course we have a little basement membrane here at the bottom of the epidermis, and the basal cell layer is connected to the basement membrane by hemidesmosomes. And if we have autoantibodies against hemidesmosomes, then this will result in a vesicle between the basal cell layer and the basement membrane, and a disease which is known as bullous pemphigoid. So again, autoantibodies against desmosome results in intraepidermal vesicles and pemphigus vulgaris, and autoantibodies against hemidesmosomes results in destructions of the hemidesmosomes which connect the basal cell layer to the basement membrane, subepidermal vesicle, and a disease known as bullous pemphigoid. Then we have the granular cell layer. It's not so prominent here, in fact, but we can see that there is a cell layer here that we have granules. That is also important for many skin conditions. If we study pathology slides, we often see if this granular cell layer is present or not. It can be absent sometimes. For example, if we have this keratosis, so some mistake in the maturation of the cells, then it gets lost. So often if I study a pathology slide, I will look for the absence or presence of the granular cell layer. Those granules are of course keratohyalin granules. And then, as you see, we started with columnar cells here at the stratum basale, and then we had the more cuboidal appearing cells in the stratum spinosum, and then the cells become squamous here at the stratum granulosum, and then the nucleus is thrown out, and we get the keratin layer here. I think that is all I wanted to say about the epidermis, so we can move on to the dermis. The dermis is the underlying tissue here, and the first thing that you notice is that the dermis and the epidermis, they have a junction. This is known as the dermal-epidermal junction, and it's kind of corrugated like this. Why is that? Well, one thing you have to be very clear about is that the blood vessels that will supply the epidermis are situated in the dermis. Here we can see vasculature. Here we can see vasculature. We can see blood vessels here in the dermis. We do not have any blood vessels here in the epidermis. Let me know if you see them. And this means that the epidermis needs to get its supply from the dermis. And then one of the reasons why we have this papillae, they are known as dermal papillae here because they are like finger-like projections, is to increase the surface area between the dermis and the epidermis, and thereby increase the nourishment actually of the epidermis. Another reason is, of course, also that the epidermis rests on the dermis, is attached to the dermis, and if we have this corrugated surface, we will have a better connection between the two. The dermis actually consists of two parts. We have the papillary dermis and the reticular dermis. In this case, if we discuss the dermis, we'll zoom out a little bit. Everywhere here you can see pink collagen fibers. The papillary dermis is here where we have the dermal papillae, and then we go all the way down and we have a vascular plexus, which is known as the subpapillary arterial plexus, which is basically, it's kind of ill-defined but I would suggest that it's this plexus. You could draw a line between those two vessels, for example, and then continue here. So this could be this subpapillary arterial plexus, and thereby the border between the papillary dermis and the lower part of the dermis, which is known as the reticular dermis. The reticular dermis is very rich in elastic fibers, and it contains the skin appendages. Skin appendages, what is that? Skin appendages means that we have hair follicles and sweat glands. This is actually a bad example for both for skin appendages. I'm looking for hair follicles. I can't see any hair follicles here. Do you have any eccrine glands? This is an eccrine gland. Eccrine glands are basically consist of simple columnar cells. Simple columnar cells that can either be dark cells produce a protein, or clear cells that produce the watery part of the eccrine sweat glands. Eccrine sweat glands actually they secrete watery sweat, so this is why the majority of the cells are clear cells, which produce the watery sweat, and then we have protein, which is secreted by more darker cells, and then we have a thin myoepithelial layer around, and those myoepithelial cells contract, which then leads to the secretion of the watery sweat, and then we have ducts which actually connect those glands to the skin surface. One thing that I want to mention is Clark level. Clark level is one of the histologic parameters associated with the prognosis of melanoma. So basically, if we have a melanoma, we look at how deep the melanoma cells are infiltrating, and then give that a Clark level. There's Clark level 1 to 5. A Clark level 1 basically means in situ. In situ means that the melanocytes are in the epidermis only. Clark level 5 means that the melanocytes or the melanoma are invading the subcutis. That means that Clark 2 and 4 have to be somewhere in between. This is where the confusing parts occurred. So if we start again, Clark 1 means that the melanocytes are confined to the epidermis or in situ. Clark 2 means that the melanocytes show partial invasion of the papillary dermis, but they don't fill out the papillary dermis. They don't go to that vascular plexus yet. Clark 3 means that they fill out the papillary dermis totally. They reach this vascular plexus, which is located between the papillary dermis and the reticular dermis, and they may even have a little pushing border without actually invading the reticular dermis. So this is what I said, it's kind of complicated. Clark 4 means that the melanocytes invade into the reticular dermis, and then Clark 5, as I said, they go into the subcutis. So that is that with the Clark stages. Speaking about the subcutis, this is the subcutis. We can see lots of fat. We can see vessels, bigger vessels here. This is an artery. This is a vein. Fat cells look like this. They have, this is a fat cell. They have an eccentrically or peripherally located nucleus here. And then it's basically an empty space. The fat droplet that used to be here, it's a unilocular fat cell, a mature fat cell. But as I said, we throw the tissue through processing, and this one of the processing steps includes dehydration, where we throw alcohol on the tissue, and this washes out the fat cell, leaving us with this artifact of an empty space, which is why fat cells look like this. The cytoplasm is basically, we have very little cytoplasm left here next to the eccentrically located nucleus, and then we have the empty space. And then, yeah, we have many vessels here in the subcutaneous tissue, and this is actually a fact that we make use of in the daily clinical practice by administering, for example, subcutaneous drugs. Insulin, most famously, is injected in the subcutaneous tissue, where it then it can go to the vasculature here and be absorbed. Thank you for watching.